Hello friends, welcome to my YouTube channel. Today I will discuss about protein. So let's start. Here proteins we are the basis of structure and function of life. So let's start from the context. Today I will discuss about introduction of protein, function of protein, composition of proteins, structure of proteins, denaturation and renaturation of protein, degradation of protein. So start from the introduction. Let's use the laser pointer. We are the basis of structure and function of life. We means here the protein, composed of 20 amino acids, the building blocks, organized into primary, secondary, tertiary, and quaternary structure, classified as simple, conjugated, and derived proteins. So I have just mentioned here 4 points you can note down, and the basic introduction is that the protein is the most abundant organic molecules in our living system. It can occur every part of our cell, and it constitutes 50% dry weight, and it is the fundamental basis of structure and function of life. So next, see from the function. There are more than there are more different types of the functions or specialized function are present in the protein. So I have just broadly categorized into two parts. One is static function and there is dynamic function. In the case of static function includes this collagen and elastin that is found in bone matrix, vascular system, etc. And there is alpha keratin that is present in epidermal tissue. Dynamic function, the another name of the static function. In the case of static function, we can say this is also responsible for the structure and the strength of our body. And in the case of dynamic function, it also accurately regarded as the working horses. So this includes the protein acting as hormones, enzymes, storage protein, membrane, receptor, immunoglobulins beside their functions in genetic control, muscle contraction, etc. So there are five elements, and that is the composition of protein. So element composition of proteins is the carbon that present 50 to 55% in protein, hydrogen 6 to 7.3%, oxygen 19 to 24%, nitrogen 13 to 19%, sulfur 0 to 4%. It is very important to know the general aspect of protein structure. So here, the linear sequence of the amino acid residues that is also determines the three-dimensional structure of proteins. The structure of the protein determines its function. So let's start from the all the structure of the protein. Let's see. So primary structure, you can see here I have given here this a linear sequence of the chain that is serine, alanine, just as an example. Serine, alanine, glutamine, valine, leucine, adenine, and here you can see this is a linear sequence of the amino acid that also attached by using peptide bond, and this type of the linear sequences of the amino acid that also joined along the polypeptide chain is also called the primary structure. Let's see the polypeptide backbone, and here you can see the methionine, and here is aspartic acid, there is leucine, and is tyrosine. There are four types of the amino acid are present here, and they lose H2O. They all lose H2O to make for making the polypeptide. Making the peptide bond. Here is the peptide bond. Here is the peptide bond. Here is the peptide bond. All the peptide bonds are. They also make this peptide bond by releasing H2O. So in this case, you can see the repeating sequence of the polypeptide chain that makes a polypeptide backbone-like structure. This is a polypeptide backbone, and you can see here there are different types of the polar and non-polar side chain that is present on the polypeptide backbone. And it is the schematic diagram of the polypeptide backbone. Let's see the structure. Here you can see this is the primary structure, secondary, tertiary, and quaternary structure are present. In this diagrammatic structure, you can see this is the primary structure, that is the linear sequence of the amino acid are present here. And this, in the case of primary, they twisted itself and in a helical shape, and this make alpha helical shape. And this is the secondary structure, and here is the tertiary quaternary structure. I will discuss about the structure. But before discussing the structure, I want to say some property of the primary structure. The case of primary structure, it also written from the left side to right side. The sequence of the primary, in the case of primary structure, also written from left side to right side. In the case of left side, this also present the amino group, and there's a right side that also present the carboxyl group. And this is though there is no dissociable bond is present there on the polypeptide chain. So there is no dissociable protein is present. So the charge only due to the presence of amino group, carboxylic group, and the side chain residues. And the protein can migrate in an electric field in the presence of the charge, that is net charge, that at a specific pH. And if you think that is a positive protein, that is called cation, that is moved to the cathode, and the negative protein, that is called anion, that's moved to the anode. And it was some property or some features of primary structure. And let's start from the secondary structure. So you can see there is the alpha helix and the beta sheet structure present. Let's see one by one. From the alpha helix, you can see this is the picture of alpha helix. There's a helical structure, and in this structure you can see the all the sorry, this is the NH group, and there's a carbonyl group that also present here, and they make a hydrogen bond between them. And this type of the hydrogen bond is very weak, but remember that they collectively make it's very stable and very compact structure. In this case, you can see the side chain that is called is designated here. It is R. So R here also extended outwards from the central axis. Here this R is extended outwards from the central axis. Here the R is extended outwards from the central axis. You can see there are two features. One is, and they also joined by hydrogen bond between NH and carbonyl group, and another is the R, that is side chain, that's also extended outwards from the central axis. So it was the property of alpha helix. Let's see some property. You can note down this property. This alpha helix is tightly packed coiled structure, amino acid chain extended outwards from the central axis. So there is some property, you can note down this. And but here remember the hydrogen bond are individually weak, but they collectively they are strong enough to stabilize the structure. So let's move to the beta sheet. Here some information. Let's see the picture first. Here you can see the picture. This is the extended polypeptide chain, and here is another extended polypeptide chain is present. But but if you observe, you can easily identify that here is the hydrogen bond is present, and here is the hydrogen bond is present. Here also in this plane, each plane of this polypeptide chain that is also lie on the plane, and there they also connected by the hydrogen bond as before as the alpha helix. In the case of alpha helix, in the secondary structure that also present the hydrogen bond. In this case, a hydrogen bond is also present between the NH and carbonyl group. So it was the same feature, and in this case, you can see the extended polypeptide chain that is also present, and there two extended polypeptide chain is present, and they join together by hydrogen bond between NH and carbonyl group. And you can see here this polypeptide chain run in this direction, and this polypeptide chain run in this direction. So the direction is different, so it is anti-parallel beta sheet. If the direction is same, then it will be called the parallel beta sheet. So you can see this is the it is the parallel beta sheet because the direction is the same. It is the side chain, the polypeptide chain run in the same direction. So it is this type of the beta sheet is also called the parallel beta sheet. But here you can see in this case of C, the direction of the polypeptide chain that runs between different positions. So it is the uh, structure of anti-parallel beta sheet. Let's move to the tertiary structure. It is a tertiary structure diagram. You can see that the tertiary structure is the overall the three-dimensional structure. In this case, here I just give a picture here. This picture also it present a single polypeptide chain, and you have to know that there are different kinds of the interactions present in the tertiary structure. So there are different kinds of the bond that is present in the tertiary structure. In this tertiary structure, there are some bonds are present. First of all, there is, you can see this is the this bond is electrostatic, represents electrostatic interaction here, and there is a hydrogen bond is present. It is a hydrophobic interaction, and here is another hydrophobic, and here is a disulfide bond is present, and there is so much non-covalent forces are present. But here you remember that there is a there is a covalent disulfide bond is somewhat present here, and in this case there is a tertiary structure. There are different types of the bonds are present to make it a tertiary structure. So it was overall a three-dimensional structure. Let's see this character here. So you can see this is the tertiary structure, and this you can note down this character totally. Let's move. This is the quaternary structure. First of all, this is quaternary structure. We will recap. This is first of them polypeptide chain, and it is the linear sequence of the amino acid is present, and it is a polypeptide primary. This is the polypeptide primary structure of the polypeptide chain, and here you can see this is the secondary structure. This is the alpha helix and beta sheet are also present here, and in this tertiary structure you can see the 3D overall fold that is contains one alpha helix, and there is another beta sheet, and they coiled randomly. This is the tertiary structure that also makes 3D overall structure. In the case of tertiary quaternary structure, you can see this is a different types of polypeptide chain is present that are also designated as subunit. Here you can see the multi-subunit complex where each subunit is a distinct polypeptide chain. You can see this is the polypeptide 1, and polypeptide 2, and polypeptide 3. They also make quaternary structure. This is the multi-subunit. Before as the tertiary structure, here you can see they all joined together by non-covalent forces. The non-covalent forces like the hydrogen bond, the electrostatic interaction, this type of the interaction. But you can, but rarely here, covalent disulfide bond is present. So it was the some quality of uh, some information about the quaternary structure. You can note down from this, and let's move to protein folding, and you can note down this protein folding as we before read this. And here you can see the denaturation and renaturation. Denaturation means the protein can be denatured by the agents such as heat, urea, and that unfolds the polypeptide chain without causing the hydrolysis. So let's see this denaturation. You can see this is the native protein that gets a unique structure by using bond. You can see this is the native protein, and in this case, when after denaturation, you can see this it loses its structure, it loses its unique structure, and here you can see that the bond has been broken here. This bond has been broken, and this is the called denatured protein, and it is the denaturation. It's called in this type of the action is also called the denaturation, and another is renaturation. Renaturation means if the denatured protein returns to its native state after the denaturing agent is removed, the process is called renaturation. So, the example of renaturation I will give, this is the example of renaturation, that the careful denaturation is sometimes reversible because as known as renaturation. In the case of hemoglobin, undergoes a denaturation in the presence of salicylate. By removal of the salicylate, hemoglobin is returned. So it is the example of the renaturation. And in the case of agents, you can note down the agent of the physical agents and chemical agents is here. And next, move to the protein degradation. You can see this is the ubiquitin tagging, one by one ubiquitin tagging, that's make a ubiquitin chain on the protein, and it also indicate the proteasome to degrade the protein chain. So when it sends the protein chain, it can degrade this protein chain into small peptides by using ATP and release ADP plus inorganic phosphate, and it can degrade the polypeptide chain in furthermore amino acid residues and finally regenerate the ubiquitin as before for use. So let's see this information. The ubiquitin tagging is fast. Next, the protein sends this the proteasomal complex that sorry, and this proteolytic complex that is called the it is called the it is called the proteasome. This subunit is degrade the small peptide which can then further degrade into small acid precursor or the presented as the surfaces of the cells as a small peptides for the immune recognition. So here you can see this is the proteasomal degradation that present into the small peptides and which are then further degraded into amino acids precursors or the presented represented the surfaces of the cell on of the as the small peptides for the immune recognition. So it was the protein degradation process. You can note down the whole process, and it's very easy to know, easy to remember here. So our session has been completed, and thanks for watching. Keep supporting me, and please like, share, and subscribe.